My name is Rocco Longo. I'm a postdoc research fellow at the University of Tasmania with TIA. I've been here in Launceston for the past almost 3 years. My expertise are wine science, wine sensory, and chemistry. And today I'm now going to give you a presentation on one of my studies within a big project that we carry out in TIA for the past 3 years on Pinot Noir wine. The aim of this project was to see if we can find any distinctive character in terms of sensory or chemical attribute that could make Pinot Noir wines very, let's say, linked to the unique link to the geographical origin. Just to give you a bit of context, Pinot Noir in Australia is actually a very important variety. Australia produces around 53,000 tons of grapes, 5,000 hectares of vines, vineyards. It actually has a share of 10% of total red wine sales in the domestic market. Pinot Noir is not just cultivated to make red table wine, it's also grown to make sparkling wine. But Sam, in the next presentation, we talk more on that. It's actually, Australia is also a very old tradition with this variety because it's been planted for the first time in the 1830s. As you can see on the right-hand side here of the slide, you can see this bar chart. As you can see, Pinot Noir is actually just behind Shiraz, Cabernet Sauvignon, and Merlot, which are actually the 3 most popular variety in Australia. To give you some context, here in Tasmania, we have around 1,800 hectares of Pinot Noir vineyards, 7,000 tons of fruit produced, 4,500 of which are used to produce red table wines. Pinot Noir is ripe very early. For this reason, the only way you can achieve, you can let the grapes to develop a very strong, very good aroma, is to grow these grapes in a cool climate regions. Otherwise, you wouldn't give time enough for the grapes to accumulate all these aroma and flavors. So we have Pinot Noir in South Australia, Adelaide Hills, Eden Valley, Florida Peninsula. We have Pinot Noir in the New South Wales, not really that much in New South Wales, but most focus on Orange and the Canberra district, which actually is around the ACT territory. We got plenty of Pinot Noir in Victoria. Obviously, you got also some Pinot Noir in the Greater Southern West Australia, and you got plenty of Pinot Noir here in Tasmania, especially around the coast, north and south. Pinot Noir is actually a wine with a very complex aroma. Its aroma is affected, is influenced by several factors, which are very hard to control, really. These include winemaking viticulture practices, so the way you ferment the wine, the juice, the yeast you use, perhaps the way you prune the vines, and all the other environmental factors, such as soil, climate, but also the rootstock, the clone. We did a study on that. We look at the aroma composition of the wine, and we were able to say that only between 25 and 30 compounds actually have an impact, a strong impact on the aroma of the wines. As you can see from this cloud, Pinot Noir wines, when they're young, they smell strawberry, violet. The older they get, they tend to smell like cherries, plum. And again, the more we age the wine, the more comes this aroma term. And let's say very finest Pinot Noir are associated with this sort of truffle, earthy aroma. Of these volatile compounds, you've got the fermentative compounds, which are ester, alcohol, and acids, and then you go grape derivatives, which are monoterpenes and norisoprenoids. Aldehydes, really, these are compounds which come from the aging of the wine when you age on oak barrels. If you are interested, actually, you know more about that. We were able to publish this review paper. It was probably a month ago on critical review in food science and nutrition. So please feel free to have a look. If you want to know more, you can ask later. I give you some context of the research project. As I said before, this was a big research project, and today I will just talk about a small study, a small part of the study that we did in 2020. The aim of the project was to Australia benchmarking Pinot Noir to support growing making perception of quality and also to add value to the Pinot Noir supply chain. The wineries that were involved in this study were wineries that were willing to participate, number one. Then the wine that we selected were selected we were helped by master of wine commentators, and wine judges as well. The criteria was that they had to be representative of premium Pinot Noir regions. Today, my focus is on the 2020 experiment. We select 5 regions from across the country. It was Adelaide Hills from South Australia, Yarra Valley and Mornington Peninsula from Victoria, North and Southern Tasmania from the state here. So we select 3 wines from each region. The 3 wines had to be produced from a single vineyard, so each wine was produced from a single vineyard, so that we minimize, let's say, the influence from a different climate or soil. Okay, so 50 wines in total from the 2018 vintage. So what we done after was we look at the chromatic and color structure components, we look at the aroma composition, and we also did a study on the sensory profile of the wines. These were routine analysis. Chromatic and color structure components were analyzed by UV visible spectroscopy. We look at the range between 200 and 600 nanometers. These were all the attributes that we look at, from color density, color hue. These are actually attributes that describe the color. Chemical age, these are indexes that tell you how fast the wine age. And again, this will be non-bleachable. Total anthocyanic are the color pigments. Non-bleachable pigments, actually, those components which they're not affected by sulfur dioxide. We look also at aroma compounds, as I say. We look at about 30 analytes, grape-derived compound, norisoprenoids, and also some fermentation-derived compounds. We operate in SPME, so just the fiber. We analyze just the head space of the wine. We don't look at the liquid matrix. This is a first result that I can show you now on the color, chromatic and color structure components. I'm pretty sure every one of you knows how PCA works, but basically principal component analysis. Well, here you can see all the variables. Here you can see all the individuals. Basically, NT, as an example, is Northern Tasmanian wine. When you say 1, 2, and 3, that means their wines. One was a wine from a different winery, and so on for the rest of the regions. So how can you interpret these results? As an example, you see the Adelaide Hills wine sit on the right quadrants of this B plot. Here, the Adelaide Hills 2, 1, and 3. And then you look at this portion of the graph, and you see that actually these wines here are mainly driven by these attributes, which are chemical age 1, 2, and hue as well. So what these tell you, basically, that the wine from Adelaide Hills look older than the wines from the other regions. And this is also for some of the Mornington Peninsula, really, from this one, MP3. When I say a wine look older, it means that is a more red brick color compared, for example, to a wine from southern Tasmania, which may look purple. Of all these attributes, only 5 actually result to be significantly different. Here is a PCA on the results of the volatile aroma compounds. Again, you can see some sort of clustering in this PCA. Northern Tasmania 2 and 3 look better. You can see some clustering for the Yarra Valley wine. Adelaide Hills as well. This is a more complex PCA to explain really, because there are so many variables here. Only 4 volatile compounds resulted to be significantly different. These compounds were ecanoic acid, ethyl decanoate, ethyl 2-methylbutanoate, and ethyl 2-methylpropanoate. What that means that these compounds here maybe can be suggested as potential markers for Pinot Noir, to characterize Pinot Noir regionality. Apart from decanoic acid, which is a compound more associated with cheesy character, really, it doesn't have a pleasant character at the very high concentration, but at the small amount, it actually adds complexity to a wine. These compounds here, on the other hand, smell fruity, and especially ethyl decanoate has been shown to contribute to the red fruit character, cherries character of wines. And so you can see here, for example, the Northern Tasmania is actually here, is driven by all these aroma compounds. I will explain a little bit more in depth what we did a sensory analysis. This actually is a photo that we took that day. We had the pleasure to have Wes Person from AWRI, a senior sensory scientist. He helped out with this procedure. The primary profile analysis was carried out using 11 wine professionals, winemakers, wine judges. So basically what they did was generate a number of descriptors. This descriptor could be wine, could be a color attributes or aroma attributes. And the idea is actually compare our sample against a pivot wine. The pivot wine is actually a wine which is not part of the experiment, but is used to compare all the rest of the wines. And what you have to say when you apply this method is this wine smell, as an example, more strawberry than the pivot, or vice versa. Here, hopefully, is more clear in this slide here. These were the wines that we had, 15 wines, which were part of our experiment, and this is the pivot wine. The pivot wine was a multi-regional anoct wine with not any specific strong smell or flavor, really. This is to show you how it looks like. We didn't use this specific chart here, this table here. We use tablets. So the process was, we use an iPad. So as an example, you test, if you do this analysis, what you do, the panelist is asked to smell, to look at the color of the wine, to smell and test. As, for example, let's say this wine, as you can see also from the photo, has a very sort of red brick color, brown, also brown as well. So then you compare to this wine, and you put all the script here in this part of in this section of the table, and so on. As an example, you think that this wine smells more dark fruit and plum, okay, you put the attributes here. If you think it's actually hot, astringent, or whatever, sweet, bitter, less than the pivot, then it goes here. After that, we generate a table, a frequency table. This frequency table, what does it tell you? For example, you can see the 3 different sections: color, aroma, and palate. And then all the group of attributes. When I say group of attributes, means that, for example, red fruits here include strawberry, it may include cherries. We need to make these sort of clusters here. What the color, what the number here say, that means that this wine, this specific wine, for this specific wine, red color was measured 7 times, brown color 10 times, H2 10 times again. In terms of the color, you can see that the blue color is actually means that the wine was scored lower than the pivot. When you see the blue gradient, when you see the red gradient, it's actually more. So as an example, AH wine, which is Adelaide Hills Wine 1, was scored 10 times for a brown character, more than the pivot wine. This is what we come up with. The color coding is similar to what I've used for the PCA. Red is Adelaide Hills, green Mornington Peninsula, and so on. Here, when you say A, C, and P, you can see that some attributes have a C, that means it's a color attributes. When you see A, that means it's an aroma, and P for the palate. We can see some sort of clustering as well, which is quite interesting, really, especially because we don't have to forget we use commercial wines for this experiment. Potentially they've been produced using so many different winemaking techniques. What we can say for the Mornington Peninsula wine, we can say that these wines were characterized by a red fruit aroma and an acidic, sour palate attribute. This result is very interesting because what does it say? The wines from Adelaide Hills were associated with this sort of brown color, sort of red as well, but red is very similar, we can say, to brown, which actually confirm our spectrophotometric analysis. You can see actually the purple color is quite distant by these wines. For the Tasmanian wines, we didn't notice a very specific trend, but what we can say some wines were more dark fruit than others, and also were characterized by this sort of purple color here. As a conclusion, I can say this preliminary study suggests that there are some regional chemical and sensory distinctiveness when it comes to Australian Pinot Noir wine producing regions. Some trends that we observe where the wines from Adelaide Hills look older than the rest of the other wines. They have this red brick color, and this was confirmed by both chemical and sensory analysis. On the other end, we see the Mornington Peninsula wines were more associated with red fruits and acidic descriptors. Further opportunities, in my opinion, there is a clear need for a deep understanding of Pinot Noir wines that could actually be achieved by analysis of wines produced under the same winemaking protocols. By doing that, what we're doing, we just minimize the human impact. Thank you for your attention. I want to say thanks to all collaborators that participate in this project, from Wes Pearson from WRI, Angela Mary from TIA, Mark Solomon at WRI, Luca Nicolotti, Anna Westmore, Bob Goldberg, and Fiona Kesley. I want to say also thanks to the funding body and to you that attend this small symposium. Thank you.